much for your kind introduction. I'm presenting here as the scientific coordinator of the SAFET project, the project on qualification of safety biomarkers, and I have been involved in the process of the establishment of this project since about 2.5 years ago. So in the summer 2007, some of the EFPI members had a discussion on what proposals we should bring forward. And safety biomarker qualification was one of them, and I was already involved in that phase and followed the project until its early start in June, and now I'm functioning as the scientific coordinator of this project. Some words about the company I'm representing. I'm working for AstraZeneca in Sweden. We are a pharmaceutical company with research sites in the UK, in Sweden, the US, Canada, and China and India. We focus on both small molecules and biopharmaceuticals and work in the area of cardiovascular diseases, metabolism and gastrointestinal, in the area of neuroscience, including pain research, respiratory and inflammation, oncology and infection as well. We are a huge company with 65,000 employees, and about 12,000 of those work in the research and development area. Our company spends about $5 billion per year on research and development. As Jackie already pointed out, many of the drugs fail in development, and the question is, why do they fail and how can we change that? And if you look at the graphs here, you see that toxicology and clinical safety are the main reasons for failure. And the question for us in Europe here is, how can we change that? At the moment, only 1 out of 10 compounds that starts in clinical development becomes a product, or even less. So how can we change that at an European level? One of the opportunities we see is, as we already said, in the area of developing safety biomarkers. SAFET stands for Safer and Faster Evidence-Based Translation, and the project focuses on the 3 organ areas: kidney, liver, and the vascular system. Those drug-induced toxicities are very difficult to monitor in the clinical setting today. In the kidney, about 50 to 60% of kidney function are lost at the point we are able to measure it. In the liver, we use liver enzymes and bilirubin as the current standards, but those markers are not specific and they do not predict who will recover from an injury and who will not. And in the vascular system, the problem is that there are no biomarkers to monitor drug-induced vascular injury in humans. So the goal of the SAFET project is to qualify translational safety biomarkers for monitoring of drug-induced kidney, liver, and vascular injury in humans. The consortium has 11 pharma partners, and the project coordinator is Frank Dietele from Novartis. And we have the EMEA and hopefully soon the FDA as advisors involved. And we do have 5 academic partners and 4 SMEs. And Firalis, with the leadership of Hussein Firat, has been the coordinator of the initial applicant of the expression of interest. Here's an overview on the project, and I don't know if I have a pointer. I'm sorry. So this is a very busy slide, I apologize, but I wanted to give you an overview how the project is organized. So we have a number of different work packages. The first starts, which is called the development of a generic scientific qualification strategy for translational safety biomarkers. And then we have 3 work packages regarding organ-specific toxicities. The first is on kidney injury, second on liver injury, and the fourth on vascular injury. And those work packages work separately with the identification of biomarker candidates, but follow a common qualification strategy. So the work is divided into two levels of clinical trials. The first is called our proof of translation studies, or the exploratory studies, and the winning biomarker candidates will then be fed into proof of performance studies, or confirmatory clinical studies. We have a separate work package for assay development and validation and testing of clinical samples, and the work package for integrative data analysis and establishment of a project database. And the work package 7 is on the biobank for clinical samples for qualification, and also work packages on management and dissemination and communication. Some of the facts and the status of the project. The project has a budget of about 36 million euro and is planned to be active for 5 years. We had our kick-off meeting in Stockholm in June and applied for early start of our project. So safety is actually the first IMI project that officially started. The governance structure is based on the steering committee, which each partner has one member in, and then a scientific advisory committee that supports the steering committee decisions. We have work package leaders and task leaders. And we also have an ethical committee and an IP committee active. And the project work is ongoing according to plan, and our first deliverables will be by the end of this year with a scientific biomarker qualification strategy, which we aim to publish as well. Also lists of prioritized biomarker candidates for the area of liver, kidney and muscular drug-induced injury, and clinical study templates that will be used in the qualification studies.